Dr. Archana Tiwari. Ma'am, are you there?
Good afternoon. Good morning, everyone. I'm here. I hope I'm audible.
Hello, ma'am.
Yes, I hope I'm
Yes,
audible.
you are audible.
And my slides are
Yes,
also
ma'am.
visible.
Yes, your slides are also visible, so you can please go ahead with your presentation. Thank you.
Thank you so much. Thank you. I'm Professor Archana Tiwari, and I work on microalgae, particularly diatoms. So this talk would be focused on combating age with algae. So aging is generally it includes the natural and the gradual process through which we become older at molecular level, at cellular level, and also there are so many systemic changes which are occurring in the body. So the body functions, the efficiency changes in due course of time, and the biological factors which are involved in aging include the accumulation of ROS, the telomerase shortening phenomena that we see at the cellular level, and also the mitochondrial dysfunction, which leads to reduced cellular function, and inflammation that accelerates the tissue degradation and age-related issue. So why algae and how algae can be utilized? So algae has gained a lot of attention in the coming 2 decades because they can be a natural solution to combat aging because they are rich in a lot of bioactive compounds that we find, which include antioxidants, various essential fatty acids, amino acid, nutrients, and they also help in neutralizing the oxidative stress which is generated and promote a better cellular health. So they can be utilized in anti-aging treatments, both as a nutraceutical and also as a cosmeceutical. So the algae-based supplements which are available in the market, they are basically extracted from spirulina, chlorella, hematococcus. So we have astaxanthin powders which are available in the market. Though the products are very less in number, but still these supplements, they help to combat oxidative stress, improve the skin elasticity, and also provide a lot of source of vitamins and antioxidant. And also we have spirulina based protein powders in the market. So algae derived nutraceuticals, they can be used to support healthy aging, and they can protect age-related diseases. Also, they can be beneficial in promoting better skin, better hair, and overall wellness. So one aspect is we have algae-based health supplements. Another aspect is we have different products which prevent or delay aging. Another interesting fact is they can be utilized as a good source of cosmeceuticals. So there are different algal extracts which are used in anti-aging creams, serums, body lotions. So why they are used is we can these algal extracts, they help in reducing the wrinkles, they help in better skin hydration, and also cyanobacteria, they produce UV protectants and natural compounds like MMA, which can be used as UV protectant. So that can be a natural source. The marine based algae, they have higher rate of production of metabolites. So the serums from marine algal source can be used for collagen production, for skin tightening, and also for a premature, aging that is accelerated due to pollution, stress, and UV exposure. So what are diatoms? They are a specified class of microalgae, and they contribute to 40% primary productivity. They are rich, better sources for fucoxanthin production, which is a carotenoid. We have a range of hydrocarbons which are produced from diatoms, also different phenolic compounds and flavonoids, which have great therapeutic value. So these organisms are explored for new food supplements and also as nutraceuticals. So in this study, what we have tried to do is understand what are the different products which can be synthesized from diatoms, and um, how we can enhance the productivity of these compounds. So these three species that I've mentioned, Thiocerosera phosphorus, Skeletonema monroyi, and Kitaceros gracilis, they were isolated from Vishakhapatnam and Rameshwaram coast. And when we brought the water samples to Delhi, we initially cultivated it in the standard F2SI media with a standard 100 micromoles of light, and provided them 25 degrees temperature so that the growth can be accelerated. So the water analysis was done, and we tried to mimic the similar kind of marine waste to marine seawater composition so that we could grow the diatoms. And once we could see the brown colonies on the petri dishes, we did the SEM analysis to get their ultrastructure, and the size was determined. After that, molecular studies, they confirmed the existence of these species. So the cultures were purified in the laboratory, and further we investigated the metabolites which are present in these extracts. So the pigment estimations were done using acetonic extract, and for HPLC, we use methanolic extract for putting the sample in HPLC. So the carbohydrate and the protein estimations were done using standardized method. Also, polysaccharide estimation was done. They do produce exo and endopolysaccharide, which have great therapeutic value. So the compounds which we extracted from the biomass included different phenolics, flavonoids, tocopherol, catalase assay was done, and standard protocols were used for extracting these compounds. The antioxidative scavenging activity was monitored using DPPH anti-AEC assay, and we also estimated the biomass which was left after extracting all these compounds. We just investigated whether they are capable of antimicrobial activity or not. So these are the results which shows that Skeletonema, one of the species isolated from Rameshwaram coast, Skeletonema had the highest protein productivity, while the polysaccharide and the carbohydrate concentration was also high. When we talk about phenolics, flavonoid, and tocopherol, in Skeletonema, the concentration was relatively higher. The pigment analysis, the results were more or less similar to what we got for protein and carbohydrate. So these species showed good concentration of pigments, including chlorophyll A, chlorophyll C, and total carotenoids. Major carotenoid in diatom that we find is fucoxanthine. Now, fucoxanthine is a wonderful molecule. Here we can see the HPLC chromatogram of fucoxanthine, which is extracted from Thiocerosera. So the concentration of fucoxanthine that we could achieve in the study was 18 milligrams per liter. So fucoxanthine, we find its application in the treatment of different cancers. So these are the cancers which have already been reported utilizing, the fucoxanthine, the inhibitory activity of the carotenoid pigment fucoxanthine. So different cell lines of liver cancer, colon cancer, bladder cancer, prostate cancer, skin cancer, and cervical cancer, leukemia, and breast cancer, we could see the inhibitory effect of fucoxanthine on different cell lines pertinent to these cancers. So the results were really interesting and quite promising. So when we talk about the antioxidative assay, we could observe the good TPPH and TAC activity. Also, these extracts of the carotenoids, flavonoids, they could show antimicrobial activity against 5 pathogenic bacteria, and the MIC value was also quite interesting. So in conclusion, I would like to say that marine-centric diatoms isolated from the Indian coast, they have shown to be a good source of fucoxanthine and other metabolites, and we have seen strong antioxidative and antibacterial properties of these metabolites. Uh, We can further enhance the productivity of these compounds by adaptive lab evolution, finding out sustainable sources to cultivate these microalgae, and the extracts can be used as antioxidative managing strategy and also promote cellular regeneration. So further research is on way to find out their more unique application, their cosmeceutical application, nutraceutical application, and also reducing the cost of production so that the product cost is reduced on a commercial platform. So anybody who wants to refer to the work related to diatoms, they can refer to our papers. This is a recent paper we got a few days, I think 2 days back, wherein we have reported our perspective on how tissue engineering can be achieved through algal extracts. And this is a wonderful team of my interns at Datum Research Laboratory. These students are postgraduate and undergraduate students, and they're very enthusiastic about working on diatoms. These are my postdoc and PhD students who are consistently working on exploring more applications of diatom, isolating diatoms, and rigorously involved in, interesting, innovative perspectives. I would like to thank my funding agency, ASEAN, for providing us funding to execute this work. And we are working towards addressing a lot of SDG goals, and I'm open to questions. Thank you.